Imagine a crime scene, a place where critical events unfolded, leaving behind silent, invisible witnesses. These tiny traces can tell a powerful story, helping investigators piece together what happened and who was involved. One of the most compelling pieces of evidence we often hear about in true crime is deoxyribonucleic acid, or DNA. But how exactly do forensic experts gather these crucial genetic clues? When forensic teams arrive at a crime scene, their primary goal is to identify, collect, and preserve any biological material that may contain DNA. This includes anything from blood and saliva to skin cells, hair, and even sweat. The meticulous collection process ensures that this valuable evidence remains uncontaminated and valuable for analysis, providing a unique genetic fingerprint that can link individuals to the scene of a crime. The fundamental principle behind effective DNA collection is preventing contamination and preserving the sample's integrity. Each person sheds thousands of skin cells daily, so everything must be handled with extreme care. Investigators wear personal protective equipment, such as gloves, masks, and sometimes full body suits, to avoid transferring their own DNA to the evidence or mixing it with existing samples. This strict protocol is vital because even a tiny speck of foreign DNA can compromise an entire investigation. Trained crime scene technicians utilize specific techniques depending on the type and location of the DNA. For visible fluids like blood, they might use sterile cotton swabs or absorbent paper to carefully lift the sample. If the DNA is present on a surface, such as a countertop or a weapon, swabbing with a dampened sterile swab is common. For dried samples, scraping or cutting out a small section of the material itself, like a piece of stained fabric, can be the most effective method. Each collected sample is then carefully placed into its own separate, labeled, and breathable container to prevent moisture buildup, which can degrade DNA. A critical aspect of this process is maintaining the chain of custody. Every step, from the moment a DNA sample is discovered to when it arrives at the laboratory, is meticulously documented. This includes who collected it, when, where, and who handled it afterward. This unbroken record ensures the evidence is admissible in court and that its integrity cannot be questioned. Often, presumptive tests are performed on site to quickly identify the likely presence of blood or other biological fluids before more advanced DNA analysis begins in the lab. Understanding the precision and care involved in DNA collection highlights why it is such a powerful tool in solving mysteries. By rigorously adhering to these detailed protocols, forensic scientists ensure that every tiny, silent witness can speak volumes, offering irrefutable evidence that brings clarity to complex investigations and contributes significantly to uncovering the truth.